Thank you very much, Federico. Once again, it shows really that this is hard work, and maybe the best we can get it. You can see. We move on to the last talk. It's from Eleonóra Gál, the effect of bile on pancreatic cancer, the importance of mucins.
Good afternoon. Thank you for the opportunity to be here. Today, I'm going to talk about the effect of bile on pancreatic cancer and the importance of mucins. Pancreatic cancer is one of the leading causes of mortality and usually associated with obstructive jaundice. We don't know exactly how high levels of serum bile affect the outcome of pancreatic cancer. So our aim was to characterize the effect of bile acid on carcinogenic processes using pancreatic ductal adenocarcinoma cell lines and to investigate the underlying mechanism. In the first step, we investigated the bile acids in human serums. We had three groups, control group with healthy volunteers, pancreatic ductal adenocarcinoma patients with or without obstructive jaundice. And we investigated the concentration of bile acid in human serum with high performance liquid chromatography, and we found an extremely high level in the case of a pancreatic ductal adenocarcinoma patient with obstructive jaundice. Then we pre-treated kappa-1 cell line, which is a pancreatic ductal adenocarcinoma cell lines with human serum obtained from these three groups. And we found changes in the cell morphology and the growth characteristic of the cells. So we wanted to know, is this associated with mesenchymal epithelial transmission? So we made vimentin staining. As we know, vimentin is a structural protein expressed in the mesenchymal stem cells, but not in the epithelial cells. And we found a very strong vimentin staining in the PDC-OG serum treated group, a very weak staining in the PDEC serum group, and the vimentin staining was completely negative in the control and the normal group. Then we investigated the rate of proliferation, adhesion, and viability of kappa-1 cells after the human serum treatment. We found that the PDC-OG serum significantly increased the rate of proliferation, adhesion, and increased the cell viability in kappa-1 cells. Next step, we investigated the role of bile acid in cancer progression. So we treated a normal pancreatic cell line and two pancreatic adenocarcinoma cell lines with 6 different bile acids in 2 different concentrations for 24, 48, and 72 hours. And after this bile acid treatment, we found that bile acid in a high concentration, especially at 48 or 72 hours, decreased the cell viability of the normal cells, but most of the bile acids significantly increased the rate of proliferation, adhesion, viability, migration, or the ability of invasion of both adenocarcinoma cell lines. We also investigated the colony-forming ability of these cells, and this representative picture and bar chart clearly shows that bile acids at a high concentration, especially at 72 hours, increase the number of the colonies, both adenocarcinoma cell lines, and we also investigated the size of the colonies, and we found that most of the bile acids increase the number of the colonies in the large and extra large group, which promotes larger tumor tissue development as well. As we know that bile acids play an important role in tumor progression, and we wanted to identify the underlying mechanism. We know that mucins are glycoproteins and has an important role in many cancer types. MAC4 is an oncogenic mucins and has also a very important role in pancreatic cancer, and overexpression of this isoform is associated with poor prognosis of the patient. So next step, we detected MAC4 isoform in both kappa1 and BC3 cell line, but in the normal, we didn't. Then we pre-treated these two adenocarcinoma cell lines with bile acid for 24, 48, and 72 hours. And we found that most of the bile acids time and dose dependently increase the expression of MAC4 both in mRNA and protein level. We also hierarchical clustering these mucins, and we found that 4 of the 6 bile acids made the same mucins expression level changes. And we also investigated this MAC4 in human samples and in normal pancreas. And in the case of neuroendocrine tumor, MAC4 staining was completely negative. But in the case of pancreatic ductal adenocarcinoma patients with or without obstructive jaundice, we found a very strong MAC4 staining. And we found that the overexpression of MAC4 in the PDC-OG group is associated with poor prognosis. Then we wanted to know how high serum level of bile affect the outcome of pancreatic cancer. So the 4-year survival rate of the patient shows that PDC-OG group patients have significantly lower survival compared to the PDC patients. And in the last step, we knocked down this isoform with siRNA, and we investigated also the rate of proliferation, adhesion, migration, invasion of the cancerous cells after knocking down this isoform. And we found that knockdown MAC4 increase cell death and decrease the rate of proliferation, adhesion, migration, or colony forming of the adenocarcinoma cells. So our research shows that MAC4 is a key mucoprotein in the growth of pancreatic ductal adenocarcinoma cells. So we could say bile acids decrease the viability of normal pancreatic epithelial cells, but increase the cell viability, proliferation, migration, adhesion, colony forming of the pancreatic cancer cells via the overexpression of MAC4. So taken together, we could say in pancreatic cancer patients where disease is associated with obstructive jaundice, the early treatment of biliary obstruction improves the life expectancy. And I would like to say thank you to my supervisor, Victoria Venglevelt, and thank you for your attention.
Are there any questions from the audience? I have one question. How many siRNAs did you use to knock down?
Sorry?
And how many siRNAs did you use to knock down MAC4?
The concept.
No, how many? Did you use 2, 1?
Just one.
Okay, so you need a confirmation with another one
yes.
To be sure of target.
Yes, yes.
Okay, and the last question is how do you think that the bile acids come into contact with the pancreatic cancer in the human situation? By the jaundice or on which route?
We think most of the pancreatic cancers occurs in the head of the pancreas, and maybe disclose the main duct, and the bile is just reach the cancer.
Okay.
There is some differences in the papillary cancer and the gallbladder cancer. It's the intestinal tract cancers has the HUC1, HUC2 gene, and that's a beneficial gene actually. That's a tumor suppressor. And the CD2 also, which is the very, very important sign of the intestinal type of cancer. So that's the cancer.
Yes, we investigated other mucins isoforms also.
So thank you for the session. We